going to talk to you today about viral restriction factors and how they can block the transduction of lentiviral vector. And how they can be blocked also to try to improve the therapeutic activity of the gene therapy. So as you probably all know here, gene therapy applications are expanding quickly these days with the number of clinical trials ongoing massively increasing. And they also aim to treat more and more different pathologies. They have different sort of platforms suitable for different applications, mostly two big types, the viral vectors with AAV and lentiviral vectors that aim to introduce genetic material inside the genome. And perhaps more recently, RNP and LNP who aim to, mostly through CRISPR strategy, modify the genetic information within the cell permanently to modify it. And mostly for viral vectors, production capacity actually of viral vectors can be limited for all the applications that are potentially possible. The current lentiviral vectors that are used for clinical applications, they are derived from HIV-1 virus. And to turn the virus into a viral vector, what has been done is to remove the what was deemed non-essential genes to give rise to the third generation of lentiviral vectors that compose mostly two types of gene, GAG-POL and REV, that are necessary to integrate the viral genome inside the cells. These vectors, the main property is that they are integrative, so they allow stable expression even when the cell divide. And they are currently produced by transient transfection of 4 plasmids. So the 2 plasmids I already mentioned, GAG-POL and REV. The plasmid that contains the transgene to be expressed under the control of a promoter that will be chosen, and an envelope gene. It's a glycoprotein that will be at the surface of the membrane of the particle. And this glycoprotein can be modified to change the targeting of the vector. It can also change its properties. Restriction factors, it's a series of proteins from the cells that belong to the innate immune systems. And their only common feature is that they can functionally block viral replication cycle one way or the other. So they can block the viral cycle. They are also used usually to trigger internal signaling within the cell to activate the full innate immunity in the cells on interferon pathway. And during the coevolution between host and the viruses, what happened is that some viruses, most viruses in fact, developed inhibitors of restriction factors so that they can block them and divide efficiently, replicate efficiently within cells. So in the case of HIV-1, for example, there are at least 3 proteins, VIF, VPU, and NEF, that can block different restriction factors. The consequence of this is that while the lentiviral vector were designed, the removal of what was considered the accessory proteins rendered the vectors sensitive to restriction factor to which HIV-1 is immune. So current lentiviral vectors are more sensitive to innate immunity than the normal virus. That's just a functional scheme of what happened to lentiviral vector when it enters the cell. So the particles come from the outside. There is entry of the particles either directly at the membrane or through the endosome pathway. Then the capsid is released. The viral RNA is then reverse transcribed and can be integrated. And the restriction factor can block more or less anywhere within this cycle. They can degrade the nucleic acid. They can block the opening of the capsid. They can degrade the NTPs that are necessary for reverse transcription. And what will interest is here also is that they can block the fusion step, either at the cell surface or in the endosome. And that's played by a protein called IFIN. I'm going to talk a bit more about. So IFIN proteins, it's a multigenic families in the human genome. 3 of these proteins have antiviral activities. And they're active against very different viruses, not all of them membrane. And IFIN can have different effects. I'm not going to detail them all. What will interest them mostly is that when they're expressed either on viral particle or in target cells, they can decrease fusion efficiency. So they block the fusion between viral and cell membrane. And this effect of fusion blockage can be depending on the glycoprotein that's on the virus. To study the effect of IFID protein on lentiviral vectors, I generated a series of model cell lines that overexpress each one of the IFIN protein. I'm not going to go into detail, but we checked that each cell line expressed the proteins that it was supposed to be. The protein is expressed more or less what we wanted it to, so cell membrane or endosomes. And then we devised assays to test the effect of each restriction factor. Simply, we have a GFP-marked lentiviral vector that we use to transduce each of the cell line, and the more green cells we have, the more transduction there is, so the less efficient the restriction factor. And we use this to test how if it affect lentiviral vector prototype by different envelopes. The results are here. So very basically, we tested here 3 different glycoproteins. We can see that the IFIN protein can affect differently the lentiviral vector depending on the glycoprotein that's at the surface. Cocal, for example, is nearly resistant to all the IFIN protein. And what I will insist on here is not the most impressive, but it's VSVG, because it's a protein that's commonly used for most of the clinical trial, and it's not massively, but very significantly inhibited, mostly by IFIT3. Then we studied the effect of the context of transduction, because cell line can be transduced rather easily with lentiviral vector, but for primary cell, it's usually more difficult. And along the year, people have identified compounds empirically that improve transduction. So we tested how this adjuvant can modify or not the effect of the IFID protein. I'm not going to go into detail, more or less. For each adjuvant, we calculated the fold decrease due to the restriction factor. And what we see in the graph in yellow is that some adjuvant, like polybrine and polyamine, if you don't do anything to the effect of IFID3, there's a fold decrease more or less of 4 in the case of due to IFID3. On the other hand, other adjuvants like vectofusin or cyclosporine H are able to limit the effect of the restriction factor. So they can be used to relieve the restriction due to IFID3. By intracellular staining, we showed that putting this adjuvant on the cells lead to a decrease in the total staining to IFID3. So we don't know by which mechanism exactly, but this adjuvant are able to decrease the amount of IFID3 within the cell. How does it relate to gene therapy? One of the main targets for gene therapy using lentiviral vector, actually, it's HSPC, because they are used to treat immune diseases and blood disorders, for which we need their progenitor cell capacity, which means they'll have to divide a lot, and for this reason, having an integrating vector is partly interesting. So lentiviral vector is a vector of choice. However, the CD34 plus human cells are very resistant, naturally, to transduction with lentiviral vector. And to perform clinical trial with lentiviral vector, you need a very high dose of vectors, and specific protocols have been devised to reach a therapeutic effect. So the question was, is IFID3 involved in HSPC, and can we do anything about it? So here we measured the expression level of IFID3 in human CD34 plus cells at the time where they are transduced by lentiviral vector for clinical trials. So more or less the cells are isolated, they are usually pre-stimulated, and then there's transduction protocols that can last up to 24 hours using 1 or 2 hits of lentiviral vector. And what we can see with the white bar is that at the time where the lentiviral vector is added, IFID3 is very highly expressed in these cells. And if we add cyclosporine H at the same time as a lentiviral vector, is a black bar, you see that the amount of IFID3 is very significantly decreased. And it can be correlated by the effect of transduction that's represented here. On this graph, it's a bit complicated because we compare at the same time the 1 hit and 2 hits protocol with lentiviral vector. So the dotted line is a single hit. So we just put lentiviral vector once. And we see that the addition of cyclosporine H massively increase the percentage of cells that are transduced. And we see the same increase also when we use the 2-hit protocols at the standard clinical thing. And using this condition, more than 90% of the cell can be transduced. And it's also correlated by the increase in the VCN, which is a number of viral vector integrated into the cells. So cyclosporine H managed to decrease IFIN3 level in the CD34 plus cells, and it increased the transduction. And we also showed, but it's not disclosed here, that it doesn't affect the differentiation and the engraftment properties of HSPCs. So conclusion, the model cell line we have defined on the primary cell HSPC show more or less the same response to IFID on the cyclosporine H during lentiviral transduction. We can have a model during which in HSPC we have IFID3 expressed, which blocks lentiviral vector. When we add adjuvant, whether cyclosporine H or vectofusin, we can block or limit the amount of IFID3 and allow lentiviral vector to transduce more efficiently. And this understanding of restriction factor on their effect can help us design better way to transduce cells, or at least much cheaper way, because we can gain more tenfold amount of vector to reach the same transduction efficiency, and hopefully this can be extended to other cells of therapeutic interest also. So I'd like to thank all the people in the IRTTG lab, and also CHSF maternity for all the CD4 plus cells sample, and Genopol and INSEM for funding. Thank you.
We have time for one question. No question. It was crystal clear. Okay, maybe I have one. The drug you think to use to improve transduction efficacy, may they have side effects?
Alors, cyclosporine H, no. In theory, people initially identify cyclosporine A as the effective drug. They didn't want to use it, obviously, because it's immunosuppressive, so they turned to the cyclosporine H, which is a variant that was even done in immunosuppressive. Side effect, I don't know. The engraftment studies don't show any difference when you do it in mice.
Okay.
That's all. Vectofusine, it also works. We don't know if it affects EPIN3 level, but it increased transduction. And in theory, in this case, it's a peptide that just polymerize, so we don't know. We don't expect it to be, but.
Okay. Thank you.